Hi guys, welcome to my channel. I'm Charlie. We're gonna talk about something scientific today. We're going to talk about the immunosuppression used after kidney transplant. Why talk about this topic? The reason is very simple because I just finished this assignment. The purpose of making this video is very simple, just to share my knowledge and also as a motivation video for myself in future. Something you must take note is I'm just a Year 2 biomedical science student. What I discuss here is just a little bit of the molecular biology. Molecular biology is really complicated, so I won't be able to describe everything in this single video. If I make any mistake, please correct me in the comment bar below. I will be appreciated. What I taught in this video are based on recent research paper and textbook. You may refer to the reference for further information. What is an immune system? Immune system protects us, the host, from the infection of the bacteria, pathogen. Anyhow, it's non-self. So the immune system will recognize our own self as self, and then for the other than self, it will recognize as non-self. So the problem with the non-self, it will fight against it and try to eradicate the non-self. Kidney transplantation is a surgery procedure that replaces a failed kidney with a healthy kidney from a donor. It is a treatment option to improve the quality of life of patients with end-stage kidney failure disease. HLA are surface glycoprotein found on most of the cell. HLA are unique in every individual except in identical twins due to its highly polymorphism properties. MHC is the genomic region that encoded HLA. HLA A, B, C, DP, DQ, DR are major subtypes of HLA. According to a meta-analysis of 486 recipients, HLA mismatching have a direct impact on graft rejection. Also from the same meta-analysis, HLA-DR mismatching show a significant severity of graft rejection. HLA typing was performed prior to the surgery. HLA of both donor and recipient were compared. The HLA expressed in the donor cell must at least match with 6 HLA markers expressed from the recipient to lower the chance of rejection. ABO blood group antigen, HLA and non-HLA antigen can be recognized by the immune system as non-self. Rejection is caused by preformed antibodies, cells, or antibody-mediated immune response, and long-term allo immune response. The recognition of the peptides from donor organ as foreign by the recipient's immune system, known as allo recognition. CD4 T cells, which is helper cells, are essential in allo recognition. Indirect and direct allo recognition pathway are two pathways in the allo recognition. MHC of the recipient APC process and present the donor's HLA peptide fragments to its TCR in the indirect pathway. Donor's APC present its intact HLA antigen and being recognized by the recipient TCR in the direct allo recognition pathway. The difference between direct and indirect allo recognition is in the indirect allo recognition, when the recipient's MHC was used to present donor's HLA peptides to the recipient TCR itself, while the direct allo recognition occurs when the donor's MHC used to present its HLA antigen to the recipient's TCR. T-cell activation requires 3 signals. APC present non-self peptide to the TCR through the MHC on its surface. TCR-MHC complex trigger downstream intracellular signaling pathway and this is known as signal 1. Calcium ions are essential for the subsequent calcineurin pathway in order to activate the transcription factor of interleukin-2 genes. IL-2 is then used in T-cell proliferation. CD8D or CD86 with CD28 complex and CD40 with CD154 complex are essential for the T-cell for the full T-cell activation. The binding of the co-stimulator CD28 bind to the CD8D or CD86 is known as signal 2. T-cell is now completely activated by action of signal 1 and 2. Cell cycle, chromatin remodeling, and alteration to the DNA methylation are triggered by the signal 2. These signaling pathways are important to produce new T-cell. CD8D or CD86, CD28 complex induces the production of various cytokines and its receptor. Besides, signal 2 induces production of CD40 ligand, which is needed for the thymus-dependent B-cell activation. Signal 2 results in the production of cytokines and activation of the effector pathway. Cellular cytotoxicity, humoral response, and delay type hypersensitivity reaction are the main effector pathway. Together with the signal 1 and 2 transcription of genes that is needed for the T-cell proliferation starts. Production of IL-2 is known as signal 3. IL-2 binds to its receptor and promotes T-cell proliferation. T-cell activation induces the migration of activated effector cells such as T-cell, B-cell, NK cell, macrophage to the site of antigen to neutralize the antigen. What is immunosuppressant? Immunosuppressant are the drugs that suppress the ability of the recipient's immune system from recognizing the HLA antigens from the donor organ through the action of blocking the receptors in T-cell activation and blocking the downstream or this signaling pathway. The main target of immunosuppressants are T-cell dependent immune response. There are two types of immunosuppressant will be given to the patient. Induction immunosuppressant are the antibodies is given to the patient before the transplantation and for high-risk patients. The maintenance drug is given to the patient after the transplantation to prevent chronic rejection. So what does it mean chronic? Chronic means long-term. Induction antibodies cause cell death through the complement-mediated lysis reticuloendothelial system and antibody-dependent cell-mediated cytotoxicity mechanism. Induction agents included polyclonal antibody, monoclonal antibody, and monoclonal non-depleting antibodies. Peripheral lymphocytes are circulating mature lymphocytes including T cell, NK cell, B cell. The function of these lymphocytes are eradicating foreign peptides from the body. Rabbit anti-thymocyte globulin is an antibody derived from the rabbit that targets the surface protein on the human lymphocyte. The targeted peripheral lymphocytes are eradicated through the mechanism of complement-mediated lysis removal by the action of reticuloendothelial system and ADCC. RATG induces the expansion of regulatory T cells, which is downregulate the T cell activation. The action of the RATG results in rapid clearance of the activated T cell in the peripheral circulation. There are four types of monoclonal antibody, which is anti-CD3, anti-CD52,
anti-CD20 and LEA29Y. CD3 is a protein molecule located beside TCR. CD3 transfers intracellular signaling when the MHC T-cell complex is formed. OK3 binds directly to the CD3, causes internalization of the CD3 T-cell complex. The binding of the OK3 to the CD3 leading to the complement-mediated lysis and opsonization for clearance by phagocytic cells. The absence of the CD3 interrupts the downstream signaling pathway, and thus it blocks signal 1 of the T cell activation. The risk of OK3 included cytokine release syndrome. CD52 is present on all of the blood mononuclear cells, including T cells, B lymphocytes, monocytes, macrophage, and eosinophil. The binding of the anti-CD52 monoclonal antibody to the CD52, leading to the ADCC, complement-dependent cytotoxicity (CDC), and the induction of the apoptosis. This reduces the level of the lymphocytes. The risk of anti-CD52 monoclonal antibody is infusion-related toxicities. CD20 was found on the surface of early B cell and played an important role to differentiate B cell into the plasma cell. The binding of the anti-CD20 monoclonal antibody to the CD20 caused the destruction of the B cell directly through the CDC and ADCC and indirectly via the structural changes and apoptosis. The antibody also targets B cell activating factor BAF and its receptor, which is fundamental for the B cell survival. Besides, anti-CD20 antibody target on core stimulatory molecules such as CD28, CD154, CD40 leads to decrease in the active T cell. Anti-CD20 antibodies remove the B cell and block the signal 1 of T cell activation. The risk of using anti-CD20 antibody is tachycardia. LEA29Y is a fusion protein composed of cytotoxic T cell lymphocyte-associated antigen 4, CTLA4, with the fragment crystallizable region of IgG. CTLA4 has a higher avidity binding to the CD8 or CD86 as compared to the CD28. The binding of the CD28 to the CD8 or CD86 is essential for the T cell activation. The binding of the CTLA4 to the CD8 or CD86 inhibit the core stimulatory pathway, thus inhibit the T cell activation. The action of this antibody blocks the signal 2 of the T cell activation. CD25 is interleukin-2 receptor alpha chain, which is expressed on activated T cell. IL-2 is essential for T cell proliferation and differentiation of B cell into plasma cell. Binding of the anti-CD25 monoclonal antibody to the CD25 inhibit the T cell proliferation and differentiation of B cell into the plasma cell. Binding of the CD25 antibody to the CD25 block the autocrine and paracrine survival. Anti-CD25 antibody target activated T cell. Anti-CD25 antibodies block signal 3 of the T cell activation. The risk of anti-CD25 antibodies included hypersensitivity reaction, causing urine as a phosphatase to dephosphorylate nuclear factor of activated T cell. Dephosphorylated NFAT is a transcription factor that binds to the promoter of interleukin-2, leading to the production of the interleukin-2, which is needed for the T cell proliferation. Cyclosporin A binds to the immunophilin cyclophilin. Tacrolimus binds to the FK binding protein, forming a complex. The failure of the dephosphorylation of the ENFA enables drive the transcription of IL-2, thus there is no IL-2 production. Calcineurin inhibitor is effective in interfering the action of humoral immune response. Cyclosporin A has a higher chance leading to the hyperlipidemia, which increases the risk of getting cardiovascular disease. The risk of calcineurin inhibitor included nephrotoxicity. mTOR is a serine or threonine kinase. Phosphorylation of the mTOR activates PI3K pathway, which play an important role in cell cycle progression and protein synthesis. mTOR activates ribosomal enzyme P70S6 kinase and blood inhibitory protein 4E-BP1. These steps are essential for translation of protein and progression of G1 to S phase of cell cycle. mTOR inhibitor bind to intracellular immunophilin binding protein FK506BP, forming a complex with mTOR. Signal transduction through IL-2 receptor was inhibited as a result of the binding of mTOR inhibitor to the FK506BP. G1 to S phase of cell cycle was interrupted in the absence of signal transduction from IL-2 receptor. The blocking of mTOR pathway in T cell inhibits cytokine action, cell cycle progression, and clonal expansion. Sirolimus promote the generation of Treg cells, which suppress T cell proliferation and cytokine production. Cell cycle and immune activation were interrupted. The risk of using mTOR inhibitor included pulmonary toxicity. Antimetabolite interfere the cell cycle progression and thus inhibit T cell synthesis. Azathioprine as a competitive inhibitor of purine synthesis, while MMF as a non-competitive inhibitor of purine synthesis. Azathioprine is a product that converts to the 6-MP in the liver. 6-MP have a similar structure to the purine and able to join DNA. Presence of 6-MP inhibit the formation of phosphoribosyl pyrophosphate, which is used in purine synthesis. The reduction of the purine nucleotide reduces gene transcription and interferes the cell cycle progression. The risk of azathioprine included leukopenia. Inosine 5-monophosphate dehydrogenase is a key enzyme used in guanosine nucleotide synthesis. IMPDH is reversibly inhibited by mycophenolic acid MPA, which is a metabolite of MMF. MMF is more lymphocyte specific and less myelosuppressive than azathioprine. Risks of MMF include dyspepsia. Corticosteroids affect cellular transcription, interfering with the signaling pathway and downstream kinase. Corticosteroid fat-soluble properties enable it to diffuse through the cell membrane, binding to glucocorticoid receptor, forming a complex. Heat shock protein were released from glucocorticoid receptor, and this allowed the translocation of the complex to the nucleus. The complex then binds to the transcription factor that produces cytokine. The production of cytokine IL-1, 2, 3, 6, IFN gamma, TNF alpha are decreases. The absence of cytokines failed to activate effector pathway to destroy non-self antigen. The number of CD4 plus T cells in the circulation are reduced as a result of the impairment of macrophage by corticosteroid. High dose of corticosteroid can lead to lymphocyte apoptosis, thus the number of the circulating lymphocyte decreases. Besides, corticosteroid interfere signaling pathway of surface receptor and downstream kinase. Corticosteroids are good in reduce inflammation through the action of reducing histamine prostaglandin. These two molecules used in inflammation action. Prednisone is a prodrug of prednisolone. The difference between these two drugs is prednisone requires the action of metabolism of liver to convert it to active drug in the liver. Patients with liver disease will be given prednisolone, which is an active drug, to reduce the burden of the liver. The risks of using corticosteroids included nephrotoxicity and diabetogenic. Different types of immunosuppressant work hand in hand. Few types of immunosuppressants are given to the patient at the same time.
Please subscribe to my channel if you like my video, and see you next time. Bye bye.